Kia ora team, welcome back to the 2.5 Genetic Variation and Change 4-credit external video series. This is video 9. In this video, you will learn about lethal alleles. And by the end of this lesson, you should be able to define lethal alleles, distinguish between dominant lethal alleles and recessive lethal alleles, and solve the genotype and phenotype of individuals from crosses involving lethal alleles. Let's get started. As you've learned in previous lessons, normally different alleles of a gene just changes how a trait looks. Like the 2 alleles for snapdragon flower color can create red, pink, and white phenotypes. Another example is the 4 alleles in color point restriction in cats can create cats that are fully pigmented or fully lacking any pigment, or having a range of color point restriction phenotypes. In the example of these chickens, we saw that 2 alleles for feather color in chickens can create black, white, and speckled phenotypes. And we also saw in human blood types, the 3 alleles in the ABO blood group system can create type A blood, type B blood, type AB blood, and type O blood phenotypes. But sometimes an allele can be so disruptive that it can actually cause death in the organism if that organism inherits that allele in certain combinations. These alleles are called lethal alleles, and they don't just change appearance, they affect the organism's survival. So what is a lethal allele? A lethal allele is an allele that produces a phenotypic effect that causes death in the organism. Death can occur at any stage of life, but it usually occurs early in development, so in utero in the embryonic stage, or before reproductive age. But sometimes, in some cases of lethal alleles, death occurs later in life after the organism has reproduced. Lethal alleles are caused by mutations in a gene that's essential for growth, development, and the survival of an organism. There are 2 types of lethal alleles. The first type is recessive lethal alleles, which cause death only in homozygotes. The second type are dominant lethal alleles, which cause death even if just 1 copy of the lethal allele is present. So it causes death in both homozygote and heterozygote conditions. Lethal alleles can actually have a dramatic effect on the phenotype ratios. As you'll remember, Gregor Mendel's classic P crosses gave us a 3:1 phenotypic ratio when 2 heterozygotes are crossed. 3 showing the dominant trait, and 1 showing the recessive trait. But lethal alleles disrupt this pattern because one of the genotypes doesn't survive to be observed in the offspring. This was actually how lethal alleles were discovered, through their unexpected effect on phenotype ratios. So a little bit of history as to how lethal alleles were discovered. In 1905, Lucien Queneau was studying the inheritance of coat color in mice. When he crossed 2 yellow mice together, he expected Mendel's familiar 3:1 phenotypic ratio in the offspring, but instead, he consistently observed a 2:1 ratio, 2 yellow mice for every 1 non-yellow mouse. This really puzzled him because it suggested that something unusual was happening. So he carried out some test crosses. So if we go back from what we taught you previously, test crosses are used to determine the genotype of phenotypically dominant individuals. So when he used careful test crosses, he was able to show that all of the yellow mice were heterozygotes. So despite many crosses, Queneau never produced a mouse that was homozygous yellow. A few years later, in 1910, American scientists W.E. Castle and C.C. Little confirmed Queneau's results, and this was their data. They too saw a 2:1 ratio of yellow to non-yellow mice. When they did further research, they discovered that the homozygous yellow mice that would have been in this box here actually died during embryonic development. So in other words, the yellow allele is lethal in the homozygote. This explained the strange 2:1 ratio that Queneau saw. What looked like a break from Mendelian inheritance was actually consistent with Mendel's 1:2:1 genotypic ratio, once they considered embryonic death was actually a hidden phenotype. So these discoveries were the first clear evidence of lethal alleles, genes that are so essential to development that a mutation creating a new allele can cause death. So let's start with a classic example of lethal alleles, the yellow coat color gene in mice, studied by Queno in 1905. The agouti gene A codes for the agouti signaling protein, which helps control coat color by regulating pigment distribution in a hair strand. There are two alleles for the agouti gene, A for a normal agouti coat and AY for the yellow coat. Crossing two yellow heterozygous mice gives the expected genotypic ratio of 1 to 2 to 1. But because the homozygous yellow embryos with a genotype of AY die in utero, the observable phenotypic ratio is 2 yellow to 1 normal brown agouti. Since 2 copies of the AY lethal allele is needed for lethality, we know that this is a recessive lethal allele. Death only occurs in the homozygous recessive condition. The agouti gene is so important in regulating other genes in embryo development that 2 copies of this lethal allele results in the death of the embryo before it even implants in the mouse uterus. Our next example is the Manx cat. The T-box transcription factor gene codes for the T-box protein, which is crucial for the correct formation of the parts of the embryo that gives rise to bones, muscles, other organs, as well as the notochord, which is basically the beginnings of the spine during embryonic development. There are 2 alleles for this TBXT gene, big M for the Manx mutation, and little m for the normal non-Manx allele. A Manx cat is a cat with no tail due to the heterozygous big M little m genotype for this T-box transcription factor gene. And crossing 2 Manx cats that are heterozygous for this genotype gives the expected genotypic ratio of 1 to 2 to 1. But because the homozygous embryos with a genotype of big M die in utero, the observable phenotypic ratio is 2 Manx tailless cats to 1 non-Manx cat with a normal tail. Since 2 copies of the Manx lethal allele is needed for lethality, we know that this is a recessive lethal allele because death only occurs in the homozygous condition. Before I move on to the next example, did you recognize that the Manx cats are not only an example of lethal alleles, but they're also an example of incomplete dominance? The heterozygous phenotype is a blend of the big M allele for defective spine development and the little m allele for normal spine development, since the tail is an extension of the spine. Okay, we're halfway through our examples. The next one is the rare human disease cystic fibrosis. Cystic fibrosis is caused by mutations in the CFTR gene, which produces the cystic fibrosis transmembrane conductance regulator CFTR protein. This is a chloride ion channel in epithelial cells, such as the cells in the lining of our airways, digestive tract, and skin. The gene has 2 alleles, and they are big C for normal functioning CFTR protein, and little c for a defective CFTR protein. Now, we don't perform crosses on humans because humans aren't lab animals, so this Punnett square is just hypothetical and shows us the predicted genotype of offspring from a cross between 2 heterozygotes. The expected genotype ratio is 1 to 2 to 1, but the phenotype ratio is now 3 unaffected to 1 affected by cystic fibrosis. It's important to note that of the 3 unaffected, 2 would be carriers of the lethal little c allele for the defective CFTR protein. That means that those heterozygote carriers could pass on the disease allele to their offspring. Since 2 copies of the little c lethal allele is needed for lethality, we know that this is a recessive lethal allele because death only occurs in the homozygous condition. A person with cystic fibrosis has an imbalance of chloride and water transport across their membranes, leading to thick mucus in the lungs and in the digestive tract. This causes respiratory infections and malnutrition. And according to the Cystic Fibrosis Foundation, of the cystic fibrosis deaths reported in their registry in 2024, half died before the age of 39 years old. Although there's no cure for cystic fibrosis, treatments have significantly improved the quality of life and the life expectancy of people affected by cystic fibrosis over the decades. Our next example is a relatively common genetic disease called sickle cell disease. I say it's relatively common because around 8 million people live with this disease worldwide, and it's most prevalent in sub-Saharan Africa, but it also occurs in parts of India, the Mediterranean, and the Middle East. The sickle cell disease is caused by a mutation in the HBB gene, which codes for beta-globin protein, which is a part of a larger protein complex called hemoglobin. So beta-globin protein is key to ensuring hemoglobin has the right shape to carry oxygen efficiently throughout the body. The two alleles for this gene are HA for normal hemoglobin and normal red blood cells, and also HS for a defective hemoglobin and a sickle-shaped red blood cell. So this is what a normal-shaped red blood cell and a normal-shaped hemoglobin protein looks like, whereas the defective hemoglobin forms these long strands on the inside of a red blood cell, and that alters the shape of the red blood cell, causing them to have a sickle-like shape. This Punnett square shows us the predicted genotype of offspring from a cross between two heterozygotes that are carrying one copy of the normal hemoglobin, HA allele, and one copy of the defective hemoglobin, the HS allele. The expected genotype ratio is 1 to 2 to 1, but the phenotype ratio is 3 unaffected to 1 affected by sickle cell disease. You could also argue that the phenotype ratio is actually 1 to 2 to 1, where 1 out of 4 is unaffected and only produces the normal hemoglobin. 2 out of the 4 are unaffected but produce both types of hemoglobin, the normal and the defective, at the same time. And they're also genetic carriers of the disease. So this means that the HA and HS alleles are codominant because both alleles are expressed together equally and independently in the heterozygote. And finally, 1 out of the 4 outcomes is affected by sickle cell disease because all of their hemoglobin are defective. Since 2 copies of the HS lethal allele is needed for lethality, we know that this is a recessive lethal allele because death only occurs in the homozygous condition. A person with sickle cell disease suffers from vaso-occlusion, where sticky sickle-shaped cells can pile up and clump in the blood vessels and block blood flow, leading to chronic excruciating pain. And people with sickle cell disease primarily die from complications like severe infections, organ damage, and stroke. Now, all of the conditions I've discussed so far have been examples of recessive lethal alleles, where 2 copies of the lethal allele must be present in the genotype for lethality to present in the phenotype. So in other words, the allele only causes death in homozygotes. This 5th and final example is the only detailed example I could find for dominant lethal alleles. Huntington's disease is caused by a mutation in the Huntington gene, which codes for the Huntington protein. The normal function of this Huntington protein is not well understood, other than it's involved in transport inside neurons. But we know that the mutated form of this Huntington protein is toxic in the brain, causing movement disorders, cognitive decline, and death. The 2 alleles for the Huntington gene are big H for the toxic Huntington protein that causes neuronal death, and little h for the normal Huntington protein. This Punnett square shows us the predicted genotype of offspring from a cross between 2 heterozygotes that are carrying 1 copy of the toxic Huntington allele and 1 copy of the normal Huntington allele. The expected genotype ratio is 1 to 2 to 1, but the phenotype ratio is 3 affected by Huntington's disease to 1 unaffected healthy individual. Did you notice how 3 of the squares in this Punnett square is now shaded red, whereas in the past 4 examples of recessive lethal alleles, we only ever had 1 red square in the Punnett square? That speaks to the lethality of this dominant lethal allele, where only 1 copy of the lethal allele is needed for a deadly phenotype. I found it really difficult to find other examples of dominant lethal alleles because they're very rare and alleles tend to disappear from populations really quickly. The only way for dominant lethal alleles to persist or hang around in populations is if the dominant lethal allele only shows up later in life and causes death after a reproductive age, like what we see here in Huntington's disease. So in summary, a lethal allele is an allele that produces a phenotypic effect that causes death in the organism. Death can occur at any stage of life, but it usually occurs early in development or before a reproductive age. And lethal alleles are caused by mutations in a gene that's essential for growth, development, and the survival of an organism. Finally, there are two types of lethal alleles: recessive lethal alleles, which cause death only in the homozygous condition, and dominant lethal alleles, which cause death even if just one copy is present. So that's in the homozygous and the heterozygous condition. So by now you should be able to define lethal alleles and distinguish between dominant and recessive lethal alleles. And finally, you should also be able to solve genotype and phenotype ratios from crosses involving lethal alleles. Thanks for watching, and I'll see you in the next video.